Good day, professors, blackmates, and schoolmates. We are the Group 4 of BSMLS 2A and we are tasked to perform the Model 4. The first task is the DNA chick cell extraction, while the four other tasks are to obtain the DNA of the different biological samples. The importance of proper laboratory rules is mainly to protect the individual from exposure to harmful risks.
Hello everyone! Today we're going to extract DNA from saliva, which is my biological sample.
I measured the extra water for garbling with the use of 10 cc syringe, which is equivalent to 10 ml. I repeated this step for 4 times until I was able to get 40 ml in total. Then, I gargle it for 2 minutes. After 2 minutes, I spit it out on a clean plastic cup. I added 1 teaspoon of salt into the sample. On a separate glass, I already have here the 250 ml of warm water in a liquid measuring cup. I put 5 teaspoons of dishwashing liquid, then I stirred it well. Going back to my sample, I added 2 teaspoons of my liquid dishwashing soap solution on it. I also added this pineapple juice I extracted from a canned pineapple. I stirred it gently and then poured 2 teaspoons of chilled 70% ethyl alcohol in the solution and leave it for 1 minute. After a minute, the solution started to form small particles. After 30 minutes, a little of these particles looked like a saliva. After 2 hours, these particles were scattered on my solution. It's not a saliva, it's the DNA.
Good day everyone. Today, we are going to perform an experiment in extraction of DNA to our biological sample, onion.
Cut the onion into small pieces. Get a teaspoonful and put it in a mortar. Mechanically grind it with a pestle to smash the onion. Smashing the onion will extract the juices in which it contains the DNA of the plant material. Then add a 1/2 cup of warm water and grind it again to support the extraction of juice. After grinding, pour the grinded material in a separate cup. Scoop a teaspoon of salt into the sample and mix it in. In a 250 ml of glass water, add 5 teaspoons of liquid dishwashing soap. Stir it to evenly mix it. Get the cup of your grinded onion and add 2 teaspoons of liquid soap solution. Stir it well. After mixing, get a separate cup and strain the liquid portion of your solution using the strainer. Afterwards, add Add 2 drops of fresh pineapple juice into your sample. Stir it again to proportionally mix it. And then pour 2 teaspoons of 70% chilled ethyl alcohol. Observe the solution after 1 minute, 30 minutes, and 2 hours.
Dear professors and classmates, today I will be performing an experiment.
Before carrying out the experiment, we must gather all the necessary items to ensure the experiment proceeds successfully. Since fresh pineapple was unavailable, canned pineapple was utilized instead. The next step is to squeeze the pineapple slices to extract juice to be used later in the experiment. The banana was sliced into tiny pieces, making it easier to mash without the use of mortar and pestle. Place a 1 teaspoonful of banana in a bowl and begin mashing it slowly. A fork was used to grind the banana slices because it is the only thing that is available. Add a half cup of warm water to the bowl and continue mashing the banana until you can see that the juice of the banana was mixed in the water. And transfer it into another cup. Add a 1 teaspoon of salt to the sample and observe the changes that occur after the salt is added. Pour 250 ml of water into another cup, then add 5 teaspoons of concentrated dishwashing liquid. Stir everything together thoroughly. Scoop 2 teaspoons of that solution and add it to your sample, then mix it slowly. Add 2 drops of pineapple juice after that. Because there was no dropper at home, a syringe was used. Slowly stir it in. Lastly, get the 70% ethyl alcohol chilled within 30 minutes and add 2 teaspoons of it to your sample. After that, observe the changes that will happen to your sample after a minute, 30 minutes, and 2 hours respectively.
Good day! I am Christine Baltazar and I am taking Bachelor of Science in Medical Laboratory Science. So today, I will be performing my laboratory experiment in our biochemistry class.
For the first step, skin a freshly cut ripe papaya. Carefully cut it into tiny pieces to make the grinding process easier. Then scoop a teaspoonful of papaya and put it in a mortar, ready for grinding. Use the mortar and pestle to mechanically grind the plant material. Prepare a 1/2 cup of warm water to be used in grinding. While doing that process, pour enough water in the sample and resume grinding. Pour again the excess water into the sample. In a separate cup, pour the ground papaya. Scoop a teaspoonful of salt and put it into the sample. Observe the reaction of the salt and the sample. Prepare a separate glass for the 250 ml of water. Next, add 5 teaspoons of dishwashing liquid into the water. And with the use of a stirring rod, mix it well. Put 2 teaspoons of liquid soap solution into the sample. Use a stirring rod to mix the solution well. With the use of a strainer, carefully separate the liquid portion of the sample. Subsequently, 2 drops of juice from a squeezed canned pineapple are added into the sample. Next, slowly mix it using a toothpick. Prepare 2 teaspoons of 70% chilled ethyl alcohol into the cup. And observe the reaction.
Good day, I'll be doing our first biochemistry laboratory experiment in nucleic acid, in which I'll be using tomato as my biological sample.
Before proceeding to the experiment, we should first mechanically grind the pineapple to obtain their juice. The juice will be used in the last part of the experiment because it will help unfold the DNA. The sample of the tomato will be acquired by cutting it into smaller pieces. Then, get 1 teaspoon of sliced tomato and place it in a mortar for grinding. While grinding, pour 1/2 cup of warm water in the plant sample. Grinding will enable the cell walls to be broken down. After grinding, place the plant sample in a clear container and add 1 teaspoon of salt. Salt helps to neutralize DNA, resulting in it to become less hydrophilic. Pour 250 ml of water. Add 5 teaspoons of liquid dishwashing soap and stir in a separate glass. We are now ready to add 2 teaspoons of liquid soap. Liquid soap contains a component called sodium lauryl sulfate that can remove fats and proteins. It enables the removal of the cell membrane because they are made of fats and proteins. As a result, the DNA will go out of the cell. Then add the pineapple juice that we grind in the first part. It will help in the removal of proteins so that we will only get the DNA. Lastly, add the chilled 70% ethyl alcohol to inhibit the precipitation of the DNA. The results of the samples after a minute, 30 minutes, and 2 hours were as follows. After a minute, there were no significant changes in all of the samples. After 30 minutes, we can see that there are structures of particles. The DNA from the samples was quite evident after a few hours. The particles have already begun to cluster together, especially after 14 hours. The particles merged to form a single large structure. After performing an experiment, it is also important for us to know how to properly dispose the materials used in order not to impose danger to other people. Also, we should remove our PPE properly. We should take off our gloves first, then the face shield, and finally we'll take off our lab coat. Again, we are Group 4 and we thank you for lending your time to watch our experiment.